Hey kids, have you ever wondered what happens in the humoral response pathway? Well, let's go deep inside the human body and find out. The activation phase begins when an invading non-self bacteria is phagocyzed or engulfed by a macrophage.
Yo, I'm a non-self bacteria. I secrete foreign molecules called antigens that aren't normal to this organism.
Hi, I'm a macrophage. I'm going to eat you.
What? No, why?
Because I don't recognize you as part of this body and a self-cell.
And inside the lysosome, inside the macrophage, a lysosome containing digestive enzymes combines with the phagocytes to process the antigens.
I feel weird.
I'm the lysosome, and when I'm done with you, you feel like a whole new person.
The processed antigens combine with the MHC class II proteins, which are made by dendritic cells, macrophages, and B cells, and are presented on the surface of the APC, the antigen presenting cell.
You want to go outside, winky face? I'll take you to the surface of the cell so you can hook up with some T cells.
Looks like our little bacteria is single and ready to mingle. Helper T cells recognize the displayed antigen on the APC and bind to the MHC class II antigenic peptide complex.
Yo girl, I'm the T cell. You can call me T-Zizzle. You must be new here. What's up?
Not much. I'm just chilling. I'm pretty sure it's pretty stuffy in here.
Yeah.
Wait, hold on. I gotta sneeze.
This binding triggers the APC to release cytokinins that activate the helper T cell.
Ew.
I'm going all over the place.
The activated helper T cell releases other cytokinins that stimulate the T cell to proliferate, divide, and multiply. Thus, this produces many helper T cells, each with a receptor specific for the original processed antigen.
Ah, alright, it's time to divide. I can't take all the snot alone. There's so many of me now.
The effector phase begins when a B cell that exhibits on its surface a receptor specific for the same antigen, originally engulfed by the APC, binds to the bacteria.
You're the perfect match. I've been waiting for you all my life. You're like the sister I never had. I'm going to eat you now. I'm sorry.
I mean, okay. I don't think that's what normal siblings do, though.
The B cell engulfs the complex by receptor-mediated endocytosis. The phagosome containing the antigen fuses with the lysosome, and the antibody is processed.
I feel like I've been here before. Oh no, not this again.
I'm the lysosome, and when I'm done with you, you feel like a whole new person.
Wow, our little bacteria sure is getting busy. The processed antigen binds MHC class II protein and is displayed on the surface of the B cell.
Do you want to go outside, winky face? I'll take you to the surface of the cell so you can hook up with some T cells.
Helper T cells now bind to the displayed antigen on the surface of the B cell, causing the helper T cell to release cytokinins.
Let me bind to you, sir.
Go for it, big boy.
Achoo!
Baby, I'm back!
The cytokinins stimulate the B cell to divide and proliferate into identical B cell copies.
All right, I need to divide. Here I go.
I'm the new sheriff in town. You can call me the plasma police. I'll train my antibody troops well to kill this dang bacteria.
I'm the new librarian, and I'm going to store all of this in my memory. You can call me the memory master.
The plasma cells release antibodies with a specificity identical to that of the surface receptors on the parent B cell. These carry out the ultimate goal of fighting the foreign invaders. The released antibodies bind in a lock and key fashion to the antigen on the surface of the original bacterial invaders. This makes it easier for killer cells to attack and destroy the bacteria by phagocytosis, and the release of proteins causes the direct lysis of bacteria.
Alright, it's time to meet your maker.
No, melting.
The end.
All right, stand up front, please. Yep, you can clap. Go ahead. Does the audience have questions before I throw out some questions? Go ahead.
Where does the IL-2 come into this pathway?
Well, as we mentioned before, the IL-2 is like a specific type of cytokine.
Okay.
So when I come in, I'm like the IL-2.
Okay.
And one thing I want to clarify, I think Kaya was representing the same type of pathogen, but different individuals of the exact same species and type of pathogen. Is that correct?
Yeah.
So it wasn't that that single pathogen encounters all of those things, because it would have died each time, right?
Yeah.
So at the top, where it says macrophage ingests the pathogen or B cell ingests the pathogen, those are two different ways to begin this antibody-mediated pathway. But you could also have it begin from the cell-mediated pathway, encountering it through that process, like through the macrophage, and then have the helper T sending cytokines and then recruiting the B cell pathway. So both of these are linked. Both these pathways, it's really important to note that they're linked by the helper T. The helper T is the general of the whole thing. All right, questions from the audience? Yeah.
So I was confused about what they were saying about, like, the cytokines were released twice. Like,
I believe I heard cytokinines, where Mr. Kinine will be really happy to know that he's in this video. Little tiny Andy Kinines. No. Didn't somebody say cytokinine up there? I did. I heard it once. That was Howard at the end. It was a cytokinine.
Maybe I read it wrong.
But go ahead. Cytokine or cytokine?
no, like, they said that, like, he came out twice, but, like, when you're looking at the list, it makes it seem like it only comes out once to activate the helper T cells, and then the helper T cells activate the B cells. So where did the cytokines come in
There's two
again?
different cytokines, so you have like two different signs.
Yeah,
First time he
Yeah.
went, he had interleukin 1, and the second time was interleukin 2. So they're both cytokines, but they're two different ones. So one like binds to the B cells to activate, and the other one just for it to replicate.
But they get released at the same time, like.
No.
No.
No, it's a different situation.
What's the general purpose of cytokines and lymphokines? Or kines, however you want to say them.
To activate the helper T cells.
Okay, so to activate, and remember these are chemical signals, right? They're going to receptors, they're causing a change in gene expression inside these cells, so it may activate the cells. These chemicals form concentration gradients, so they actually dictate movement of cells in some cases too, where there's higher cytokine concentration, the cells will start to aggregate and move up the concentration gradient. What other things do we see here with IL-1 and IL-2?
It causes them to proliferate, so there gets to be a ton of them.
So activation, proliferation, which also means, what does proliferation mean?
Divide. And it multiplies.
Divide and make more. And typically the proliferation that we see in these types of responses is what we call a clonal proliferation, meaning true mitosis, where you're getting identical copies of that specific version. Let me just reiterate what we said earlier. The T cells, and to a greater extent B cells, that are just there kind of under normal conditions when there's not an attack, they'll divide at a lower rate. But when they divide, there's some significant somatic rearrangement of the DNA, so that every cell of the immune system, all the like circulating B cells and T cells, are uniquely different from one another. And so what you're trying to do is put out as many versions of those cells as possible, so that if you get an invader, the hopes are there'll at least be one cell that's a close enough fit that it'll have an effective response. And then the better it fits, the even more effective response. And you just hope over time that, okay, when you maintain those memory cells, even if you get an invader that's slightly different, that will at least work somewhat well, right? And then there's the option for that one to go ahead and divide and proliferate with some variation. So the big thing I want to drive home is your immune system is like evolution in action. You've got variation, and then you have an invader, which is a change in the environments. And then you got the clonal expansion where you're actually selecting only one of the cells to go ahead and reproduce, while all the other ones don't reproduce. So that's classic natural selection and evolution flowchart happening inside your own immune system. And so, when people say, well, I don't believe in evolution, then you don't believe in your own immune system, because you can actually see it happening. You can see it happening in real time. You can see it in action, at least at a cellular level. Now, what other questions do we have from the audience? Theodore? Liam?
Liam.
Come on, Liam. Posen?
No, I'm good.
This link is going to Hopkins.
Okay.
Impressive. Really? Last chance?
I'll go. Can
Yeah.
you go over what CD, like how CD4 gets its writing again?
See what?
CD4.
Well, that was kind of like what my question was before in the last video. Mr. Erdmann was talking about CD8, like enhances the binding of the antigens present on the surface of the cell. So it's, in yours it was CD8 that.
So are they like analogous? Like, do they do the same thing?
I think so.
I think we almost need to go see a picture, and that's what we'll do as soon as we shut down this video. I'll show you what that arrangement looks like. Okay, any other questions? All right, thank you.